Hi, I'm Professor Matthew Schmidt, and I'd like to welcome you to genetics. In this section, we're going to discuss once again linkage and gene mapping, but specifically we're going to talk about the way we can do this with certain types of fungi, and we can simplify the analysis on the one hand, and we can do some things that you really can't do any other way. So who knew fungi were so important in genetics? You do. So let's take a look at the concept of I could be wrong, but a lot of people find themselves a little bit in the dark when it comes to fungal life cycles and genetics and things like that. They are different in certain ways than animals and plants, and I mean not just the obvious ways. So certain fungi that are technically in the division Ascomycota, um, they're in that division because an ascus is a little sac in which they make their sexually generated spores. So they make these sacs of either 4 to 8 spores, and we can use them for a particularly powerful type of genetic mapping. It's called tetrad analysis. And we'll see more about tetrads in a minute and why this is so powerful. What a tetrad is, perhaps obviously, we've used that term before, but it's a little different here. Tetrad generally means 4, but there are 4 spores in one ascus, and it's not a coincidence because remember, in meiosis, generally speaking, one starting cell leads to 4 haploid gametes. In this case, they're not really gametes because of aspects of the fungal life cycle that we'll discuss very briefly, but what they are spores, and there are 4 spores in one ascus, and this is very important. That represents the product of one single meiosis event. In that little ascus, that little sac, the 4 spores that are in there came from one meiosis event. So just think for 1 second about what we've been doing. If we do mapping, say in Drosophila, we're actually counting offspring, right? And those offspring are representative of certain combinations of gametes going together. But the reason the numbers never match exactly of like the parental classes, etc., is because, you know, meiosis is going on in hundreds, if not thousands and millions of cells, and I mean it depends on the organism, but ridiculous amounts of meiosis is going on, and then all the gametes sort of end up in one pool, and they randomly sort of connect with another gamete. So we're getting an estimate, which is fine, but here we actually can tell, gee, those 4 cells, spores, came from one meiosis event. It may not be obvious why that's powerful right now, but it will be in a minute. So Neurospora is a mold, and the word mold is a little odd because technically it doesn't really mean that much, but Neurospora is famous in genetics because you get this ascus, but then an additional mitotic division occurs. This is probably just to make more spores numerically so that the fungus can be more prolific. But so technically this is called an octad, which means 8 spores, but really think of them as 4 because there's sort of 2 identical ones and then the next 2 identical ones, etc. What's very important about Neurospora is that they are actually ordered. Now, there's a lot of technicalities that go on with this. And by the way, your class may or may not even broach this subject. Some people like fungal mapping and fungal genetics. Some people shy away from it. So obviously you'll only be watching this if it's relevant to you. But the bottom line here is if you have ordered spores in an octad, you can do something called centromere mapping, and there's really no other good way to do it. So that's something I'd like to explore, and if it's something you have to do, I guess you'll be glad that we explored it. Okay? So why is this so amazing? Well, a couple of things. If you can, instead of counting offspring, for example, like we do in fruit flies, you can actually look at the spores directly inside of an ascus, and you can make sometimes direct observations of spore phenotypes. Now, what might they be? The spores could be different colors, they could be different shapes. It doesn't really matter for our purposes. But one interesting thing is that spores are haploid, right? They're the product of meiosis. So since they are haploid, the genotype equals the phenotype. We don't have to worry about any dominance effects or things like that. So not only, so I have a picture down here where I made it up, but big A would equal a yellow color, little a would equal a green color, and you'd be able to look right under the microscope at that and see that that was the case for those 4 spores that came from one meiosis event. Okay? Now, we're definitely not going to get into this next part. It's rather technical. I just want you to be aware in case your class mentions it. The fact that meiosis occurs during the 4-strand stage of meiosis, where the tetrads are, we've talked about the fact that it does occur that way. The way it was actually proven that that was the case was using fungal genetics and actually using this type of analysis with the tetrads and the spores. All right, so I don't want to say don't worry about it. It depends strongly on your professor. It's beyond the scope of this discussion to talk about that, but that was the evidence that really was the proof, the strong evidence that meiosis occurs at the 4-strand stage. Before we can do this, I think it's a good idea, before we can see how we can use this for mapping, we should take a little look at the generalized life cycle of a fungus, because it really is a bit odd compared with even plants. A lot of people think fungi and plants are similar, but they really have very little to do with one another. So here's a rather beautiful picture, I think. And since, it's a life cycle, we can start anywhere, but why don't we start here, because we just had a similar picture there. That's an ascus that has 4 spores in it. 2 happen to be capital A, 2 happen to be small a. Right now, it really doesn't matter what those 2 things mean. But a spore is something, if this was Neurospora, right, you'd get this mitotic division, and I didn't bother writing the letters of the alleles in there, but now we have 8 spores in the ascus, right? Now, the whole point of a spore, the word spore, I don't want to go into it crazily, but it's used in a little bit of a different sense, whether you're talking about fungi or bacteria or plants. In this case, the point of a fungal spore is basically to either fly in the wind or float on the water, and basically it's going to find a nice place and germinate. So in most cases, you have these haploid spores that have been released out into the environment, and when they germinate, most fungi are multicellular creatures, and the product of that germination is something we wrote here. It's the mycelium is the body of the fungus, if you could think of it that way. But each of these little strands is called a hypha. All right? A hypha is literally, sorry I can't show you one, but it's like a hair in terms of its width, and can grow very, very long. So obviously it has nothing to do with the human hair really, but that's about the width that we're talking about. So one of those strands is called a hypha. The net sum of all the hypha are called the mycelium, which is the fungal body. Okay? Now, there are different mating types in fungi, and in the most simple version, it's a little bit of a stretch to call them male and female. But it's that's the idea, that if a mycelium that's of one mating type sort of literally runs into a mycelium or a hypha of another mating type, those cells can fuse, and since they're both haploid, this is a little odd. Note that the spore germinated and it was still haploid, and all this is all haploid. Fungi are haploid dominant creatures, which is the opposite of most plants and animals. So if 2 compatible strands come together, they can fuse, and what you end up with is a diploid zygote. Now, I just drew it as if it's one cell. Sometimes it is one cell where the 2 hyphae come together.